Good afternoon. I'm very happy to introduce to you Oren Shriki. Oren is a graduate of the ICNC. He did his undergraduate studies right here at the University in physics. Then he did his PhD with Chaim Polninsky. And then he left the world of research to educate the young at the high school right here in Jerusalem, where he served as a teacher, physics teacher for 9 years. And 2 years ago, he decided to get back pursuing research and took a postdoc position at the section on critical brain dynamics at the National Institute of Mental Health in Bethesda, Maryland, USA. Owen is responsible for practically all the material we students enjoy here at the ICNC. He was TAing multiple courses and was and still is considered the best TA that served here, I think, to my opinion.
Okay. Thank you very much for the warm introduction. Thanks for hosting me, and thank you all for coming this afternoon to the seminar. And indeed, there are many buzzwords here in the title, but I'll explain all of them. What are neuronal avalanches and what's MEG? The human brain, I guess you know. So everything will become clear. And feel free to ask and interrupt during the talk. So the lab I work at the NIMH is actually a wet lab. The expertise of the lab is cortical cultures, organotypic cultures, also rats. They do calcium imaging, optogenetics, and also recordings from monkeys with arrays. But I'm applying tools that were developed in the lab and other tools that I developed on the way to MEG. And we get similar results. So it's kind of interesting, and I want to show you how we do that. I will first introduce the concept of criticality, which is important in this talk, and a signature of criticality, a specific signature, which we call neuronal avalanches. So it's not the whole story of criticality. It's a part of it. I'm telling the truth, maybe not the whole truth, for didactical reasons. Then I will explain the recipe, how we do the avalanche analysis, so it will be very clear. And I guess that after this part, you will all be able to apply to your own data. Local field potentials, EEG, MEG. There are even some works on avalanches in fMRI, but I'm more critical about them. But spiking data also. So it's not a hard analysis, and it can give some insights. I hope I'll convince you. But I will describe it in the context of MEG, show you the results, and then just summary and discussion. And I hope I'll have time to tell you a bit about the future plans. So the concept of criticality applies to dynamical systems. And we say that a dynamical system is critical when it operates on the border between two different types of behavior. For instance, in the context of neuronal networks in the brain, we can talk about the transition, we can talk about propagation of activity and about the transition from premature termination, when activity just dies out. You start activity, but doesn't really propagate, to the other extreme, where activity propagates too much. It just explodes. You get exponential blow-up of activity, like an epileptic seizure. So this is one type of criticality. There are others. But this is the one which is relatively simple, and the one which we will address in this talk. In general, critical systems have some universal properties. They have systems that do not, these systems, they have some characteristics that do not depend on the specific microscopic details. They are emergent properties of the system that depend on properties of the interactions and some global properties. So this is nice. I mean, you can tell a lot without having to know all the details. And criticality is also associated with optimal information processing. It depends on the context. This is a very broad term to say optical information processing, but it can be high sensitivity to external inputs in the context of sensory representation. It can be a larger repertoire of representations in the context of memory and also in sensory processing. I mean, there are some experimental works from our lab. I also did a lot of work during the end of my PhD and later on about criticality in a slightly different context, but in modeling, motivated by optimal information processing. Now, there is evidence that suggests that the cortex operates near criticality. Even more specifically, we talk about the superficial layers. Most of the evidence comes from the cortical cultures experiments, but also from anesthetized rats and monkey experiments, awake monkeys that were done in the lab. But all of these were done with microelectrode arrays on the scale of about 1 millimeter, and no evidence currently, there is no evidence in the context of the large-scale human brain behavior. And this is what we're out to explore in this project. So, as I said, I focus on a specific signature neuronal avalanches. An important framework that we will use in this talk is that of branching processes. So the basic context is that we discuss propagation of activity in the brain. Branching processes is a very useful language, mathematical framework to discuss such cascades of activity. It was invented originally in the context of genealogy, in the context of family names. Why do some family names disappear, others survive for longer periods? And there are many theorems and a very extended mathematical framework, which was, for instance, very useful in the context of the Manhattan Project, because nuclear chain reactions are also a type of branching process. In our case, we imagine a group of neurons that can activate other neurons. If those neurons are at a relative synchrony, then they can activate other neurons down the road, and if they activate the whole group, then there is a chance that you'll get further activation down the road, and it can propagate. But we also know that there is stochasticity in the system, so sometimes you can get short cascades, sometimes you can get long cascades. The number of events in a cascade is called the cascade size. I will show it concretely in the context of the MEG, so you will know exactly what I mean when I say cascade size in that context. Another important concept is the branching parameter, which discusses or describes the amplification in the system. For instance, here, if in the first step we have 4 active neurons, and in the next step we have only 1 neuron, then the ratio is just 1/4. But if these 4 neurons activate 4 neurons, the ratio is just 4/4. So that's simply the branching ratio, and of course we look at the average quantity in the system over a long time course. Now, we say that the system is critical when this branching parameter is 1. If it's larger than 1, then you get explosion, typically, because activity just blows up. Each time you get more and more active units. If sigma is smaller than 1, the branching parameter, then activity dies out. So these are the two extreme scenarios, and in between, activity is maintained, but again, because of stochasticity, sometimes you get short cascades, sometimes you get longer cascades. I want to explain or emphasize more the point of stochasticity. So just for example, imagine that there is a neuron here, single neuron, that can activate this neuron with probability p and this neuron with probability 1 minus p. So in principle, this can be independent probabilities, but let us assume that they sum up to 1. So then the system is critical. So on average, it will activate one of them. But maybe, for instance, here it activated both. But then this one would not activate others. It could be. So if we keep in each stage the sum of the probabilities to be around 1, then the system is critical. But the specific pattern that you will see depends on some randomness in the system. Now, of course, in general, you can have many neurons in each step, and you can have many probabilities that are different. But if they all sum up to 1, in the sense that you don't get this blow-up, then the system is critical. So we reserve the term neuronal avalanches to describe cascades when the system is critical. And we have a very important prediction of the theory of critical branching processes that appears, for instance, in this famous book in the context of branching processes and other papers, is that if the system is critical, you will see that the cascade sizes are distributed like a power law with a specific exponent of -1.5. So it comes from relatively elaborated theory. That's the prediction. Now, Okay, here's a simple example of a supercritical branching process. That's the analogy of an epileptic seizure. So here are some results. I will explain exactly how we obtained them. At this point, I'm just saying qualitatively, these are sizes of cascades. This is the density. It's a log-log scale. And these are results from an organotypic culture of Cortex from rat. And you see a power law until a certain cutoff, which is at about the system size, the number of electrodes you have in the array. And there's a reference power law here with a negative 1.5 slope, and you see that they're roughly parallel. What's interesting is that in the cortical cultures, you can also apply some pharmacological manipulations. For instance, you can block NMDA and AMPA receptors. So when you block excitation, you get what we call subcritical behavior. So you see it's more like an exponential fall, and you don't see this power law behavior. And on the other end, if you block inhibition with picrotexin, the system becomes excitable, and you get many large cascades, more than you would expect from the power law. So you get something like a bimodal distribution. This is like a signature of a system that is more epileptic. And we have similar results with electrode arrays in the monkey, prefrontal and premotor cortex. And another results which I'll also show in the context of the MEG is that if we take subsections of the array, namely fewer number of sensors, we get an earlier cutoff. So now I want to explain to you exactly how we obtain those kinds of figures. And the main question is, okay, we see it at the 1 millimeter scale, but if we have a power law reflects some scale-free behavior. It doesn't have a scale. So we expect to see it at larger scales. For instance, in large-scale human brain activity, and we have a lot of MEG data sets, resting data sets that I'm analyzing in this context. The paper is currently under review. So briefly about the MEG technology. The measurements that you get are very similar to EEG. There are some advantages and disadvantages. The main disadvantage is that it's much more expensive than EEG. It measures the magnetic signals of the brain that are associated with the neuronal current. They're roughly 8 orders of magnitude weaker than the ambient magnetic field, so you need to do everything in a shielded room, and you have very, very sensitive sensors. They're so sensitive that even makeup, I mean, they ask for instance, ladies to take all the makeup before they come. When they come, they don't have makeup, but they still force them to use a wet wipe to clean up the remains of the makeup because it's so sensitive. There are some metals in the makeup, and you can actually detect them. Now, the main signal comes from the superficial layers of cortex. It's related to the geometry of the apical dendrites that are aligned. It has a temporal resolution similar to that of EEG. In general, although not always, better spatial resolution if you try to do source reconstruction, and it's reference-free. And another advantage is that you don't need to stick electrodes. You just put your head, you raise the chair, and you start to record. So we have an MEG facility at the NIMH led by Richard Coppola. It's a Canadian machine, CTF, a very good machine, but they are bankrupt now. I won't get into more details. In most MEG machines, you have gradiometers, which measure the gradient of the magnetic field, because then you subtract, or you don't have an effect from the mean ambient magnetic field. That's why I use this gradiometer. But for instance, in Bar-Ilan University, they have just magnometers and another reference measurement for subtraction. So we have, it's NIH, so it's very easy to get a data set of 104 subjects. Resting state activity I shot 4 minutes per subject, and 600 hertz was the sampling rate in this case. So we have all these channels, and if we look into some of these, it looks something like that. We get this kind of channel activity. What we typically do for the analysis is we subtract the mean and divide by the standard deviation, so we get the signal instead of in units of femtotesla, in units of standard deviations. So what I show here is the distribution of the signal in one subject. What do I mean? I take all the channels, 273, all the time points, 4 minutes, 600 hertz, and I just plot the distribution. These are units of standard deviation. This is a logarithmic scale. And this is what you get. The red curve is the best fit Gaussian. The main idea is that each sensor just sums up many electromagnetic sources in the brain. If those were independent, if it was just pure noise, we would expect to get Gaussian behavior. But we can see that there is a deviation. Of course, some subjects behave differently. Maybe they don't have this kind of exponential fall off here, but slightly above, slightly below. But all of them are very different from the best fit Gaussian. And you can see that at about 2.5 standard deviations or more, this deviation is quite large. So why is it important for us? Because the next stage, or what we do with the data, is we first sample it to a discrete point process. So that's, I guess, the most non-trivial step here, because we take those continuous signals and we convert them into a point process. We impose a certain threshold, and then we detect the peaks. So here I use a threshold of 3 standard deviations. It's very common to use it, but I would claim that if you use thresholds that are large enough, namely above 2.5, and not too large, like if you go for too high a threshold, you just don't get events, right? So you throw the baby with the bath. So you want to have a reasonable signal-to-noise level. That's why we take these kind of thresholds. I also did some quantitative ROC, receiver operating characteristic analysis. But these things are not very crucial for the analysis. The results remain approximately the same when you change the threshold. You just get more or less events. Okay. Also, the method in which you detect the peaks is not very crucial. Here I find the excursions beyond the threshold, and I detect the extreme point in the excursion. But if I take all local minima or use other sophisticated methods, it's pretty much the same results. Just be consistent. Here's a raster from the whole array. So you clearly see some spatiotemporal structures. It's not random. If you just take random data from uncorrelated neurons, you won't get that. And of course, some of the correlations come from real correlations in the brain, and some of them, we must remember, come from overlap of sensors. It's not really trivial to discriminate. But it's important that we are not looking at just pure synchrony. We are really trying to look, I will show you how, at spatiotemporal cascades that are extended in time. Some of them last tens of milliseconds. So if you zoom in to these avalanches, or what we later call avalanches, you will see that they have some structure in it. Here, you cannot tell a lot because this is just a one-dimensional axis, and roughly adjacent sensors have consecutive numbers, but not always, because it's a two-dimensional array. But you see that you get those kind of synchronized patterns of activity. And next, we want to segment the data into many such cascades. The way we do that is we take the point process from each channel. We just put them into one big sequence of events. And now we start to cluster it along this single dimension. The way we do that is that we choose a time scale. This is another parameter of the analysis. The first was the threshold. And I will discuss this later. So we choose this kind of bin width, delta t. It's, of course, it should be a multiple of your minimal resolution. For instance, for 600 hertz, the minimal resolution is 1.67 milliseconds. So I work in multiples of this unit. And we then look for silent periods. And everything between two silent periods is called a cascade. So for instance, here we have two cascades, one of size 5, one of size 6. This is one way to measure sizes. Just count the integer number of events from the whole array. So maybe these were 5 different sensors. Maybe there were 4 sensors, and one of them fired another event. So typically in a data set of 4 minutes, I have a few thousands of cascades. And then we segment, we get all the sizes, and we can plot the distribution. It's important to emphasize at this point that the result will be sensitive to the choice of delta t. If I choose a slightly larger delta t, then those two cascades will be concatenated because there is no silent period between them. So you get one cascade of size 11, right? So an important question later would be, okay, how do I choose my delta t? But for the moment, let us start with one delta t, which I will try to convince you later that is the best. And I will show you some results with 1 delta t, which is 2 times the minimal resolution, just 3.3 milliseconds. I want to explain why we have this kind of delta t. It's a bit technical, but I want to give some intuition. From the point of view of the MEG, the Cortex may be like a continuum sheet, but really we have a finite sampling. We have some sensor spacing, which is roughly on the order of 1 centimeter in the context of the MEG, 200 microns in the context of the MEAs. And we know that we also have some range of conduction speeds in the nervous system. So it takes time from event to propagate from one sensor to nearby sensors. And this actually sets the resolution, the basic resolution that we need. If we take a very small bin width, it means that events that belong to the same cascade will be separated because we don't see any causal relationship if we take a very small delta t. If we take a very large delta t, we concatenate events that are really independent. So there is some best delta t for doing the segmentation. Okay, so let me show you one cascade. I just chose one of the cascades from the data that had a duration of 20 milliseconds. And so it means that the delta t was 3.3. So it means that before this and after this, there was at least 3.3 milliseconds of quiescence in the array. So in this avalanche or cascade were 24 sensors. These are the waveforms, and these are the events. If I show them, if I zoom in on the 20 milliseconds, and 3 of them fired twice, so you get overall 27 events. That's the size of the avalanche. We can also look at the brain activity, just interpolation of the sensor activity. So this is slightly before the avalanche, and during the avalanche, you see this hot area, and then it dies out after the cascade. So you get some evolution of activity here, and these are the sensors that were involved in the cascade. So in this case, these sensors, they form a spatially contiguous cluster. What we see, I did some quantitative analysis, is that typically, although we use only temporal information, only proximity in time to segment the cascades, they turn out to be relatively spatially contiguous, more than what you would expect from randomness. And again, part of it may come from sensor overlap, but because these are extended in time, part of it is really some kind of propagation of the activity. This is what we believe. Here's just another example. I also detect negative peaks. Maybe this is a good time to mention that when we do the analysis on local field potentials, typically we look only at the NLFPs, the negative deflections of the local field potential. They reflect, again, some synchronous synaptic activity, and they are known to be correlated with spikes. So typically, you look at NLFPs. In the context of the MEG or EEG, it's not really justified to look at either positive or negative signals, but you get the same results. If you look only on positive or negative signals, you just have less events, fewer events, but still, you get the power laws and everything. Yes? These are the events, yes.
So what is the role of, what do you mean by delta T now, or duration? Just the events that you get by threshold.
Thanks for the question. I mean, these are the events that I get by threshold, but for some reason, I call this one cascade, and I isolated this cascade from the rest of the data. Why?
What do you mean from the rest? From something which is prior in time and later in time, or there are events here that you don't show up?
So the fact that I'm calling this a cascade depends on these two parameters. For instance, if I took a higher threshold, I would get more events, or a lower threshold. But in this context, what I show you is just another cascade of 20 milliseconds, in which there was a period of at least 3.3 milliseconds before, and at least 3.3 milliseconds after. If I took, for instance, if I demanded a different delta t, this would not be a cascade, maybe. Maybe I would concatenate it with another cascade, so you would see more events.
But within these 20 milliseconds, these are all the events?
These are all the events that happened on the array in that period. And of course, when I do the segmentation, I lose all the spatial information because I put everything into one big sequence, but then I can reconstruct. ask where the events came from. Okay.
But you took all the detectors of all the array.
Yeah, so it's a good point. A limitation of this analysis is that you assume that there is one cascade at one time. This analysis do not allow you to get simultaneous parallel cascades on the array. And I have another way, which I started to investigate, a more sophisticated clustering method that can actually, I hope, will detect parallel avalanches. But here you just assume that there's one cascade in a given time.
So the sorting here is done afterwards.
Yeah.
It has nothing to do with the location.
Right. So here's a summary of the steps. I do the peak detection in each channel, and it depends on one free parameter, the threshold parameter. Then we just forget the spatial information. We put everything into one big sequence of events, and then we segment using a certain timescale delta t. That's it. And of course, if we have free parameters, we can play with them. It doesn't mean that we have to stick to one value and see what happens. What I'm measuring?
No.
Okay. What's the meaning of these events? The physical meaning of the events. It's a good point, yeah. So the MEG measures basically synchronous activity of the neurons. You don't see directly the neurons. The MEG signal measures postsynaptic potentials. We know that the spikes generate magnetic fields that cancel each other. So what you basically measure is some synchronous synaptic activity. Now it's considered using certain calculations that you need about 50,000 simultaneously active neurons to get a detectable MEG signal. I guess that if I threshold it at an even higher level, what I get are really large groups, tens of thousands of neurons that are active simultaneously. And then these signals can propagate in the cortex. When you have such a large group of neurons active, it can activate other groups.
But if this is the logic, how you can combine positive and negative
So the sign of the magnetic field depends on many things. First, recall that the geometry is complicated. It's not a planar culture. It's the convoluted cortex. It depends whether you measure from the sulci or gyri. And it depends on the alignment of the apical dendrites and the direction of the current. So it's not that trivial to say what's the meaning of a positive or negative magnetic field. And on top of that, I subtract the mean, divide by standard. I mean, these are deflections above or below the mean magnetic field. I didn't say that this is a positive or negative magnetic field, right? So these are just large deviations in that sensors. So it reflects some kind of large synchronous activity. That's what I can say. Now, okay. So there was a study that tried to compare LSC and MEG. What they basically did, they took a monkey, they did a tactile stimulation. So they just changed the ISI, the interstimulus interval between the stimulation. It was like a point stimulation. It is well known that when the ISI is large, you get higher amplitude of response, and if you decrease the ISI, you get a smaller amplitude. So what they did, they measured using MEA. They also repeated this with MEG, and they looked at the spike activity, they looked at LFPs, and they looked at the MEG. What they found is that actually LFP and MEG were highly correlated and less correlated with the spike data. What they did basically is, I need a pen. So this was the ISI, and suppose for one ISI of 2 seconds, you say this is 1, whatever, they calibrate everything to 1. So they put all the results, whether it was LFP, MEG, or spike. And then you measure for different ISI, you get something like that. And this is from LFP. And if you plot on top of it the MEG, it looks the same, roughly the same. They are very nicely correlated, but the spike data was kind of, behave differently. Not necessarily coincide here. So there is some correlation between the LFP and MEG because both are thought to reflect some kind of synchronous synaptic activity. This is what I can tell.
Did anyone do the LSP in monkeys and MEG in the same monkeys?
I'm quite sure that this experiment I described was the same monkeys. Maybe not simultaneously. I mean,
You're talking about the MEA. MEA is not the monkey.
I said MEA? No, maybe it's I don't remember if they used the Utah array or what type, but they used a certain array to record from the monkey, an MEG from the same monkey.
You're talking now about the monkey experiment?
Yeah, this is a monkey experiment, yeah. So they compared LFP and MEG in monkey. There is not much work on that, so I think these are things that needs to be investigated. But if you read the literature on the interpretation of the MEG signal, this is the best I can tell you. I tried. Okay, so next step, we can construct the distribution of the cascade sizes, right? I showed you how we segment them. We get a few thousand of them in typical data sets of 4 minutes, and now we can plot the distribution. We just so the x-axis is the size, and in this talk, I stick to the size in terms of just the number, the integer number of events. So we get this kind of power law behavior. This is a reference power law at negative 1.5. So at least for this delta t, we get roughly power law with that exponent. And you see that there is a cutoff. And the cutoff happens close to the system size. It's related to the fact that we don't see reverberation. Typically, when we have a cascade, one sensor participates one time in a cascade. Sometimes it participates twice, but this is the rare case. So you typically don't see cascades that are larger than the size of the array. Now, of course, so that's the important result. We get this kind of power law. Now, I want to convince you with some controls that it's robust, and then we can discuss more the meaning of that. So one simple control is to do phase shuffling. Namely, you just take each channel data, you Fourier transform it, and then you just shuffle the phases of different frequency components. So you keep the same power spectrum, and then you reconstruct the signal. So you destroy the temporal correlations and the temporal structure in each channel, but you keep the same power spectrum. And the power spectrum, whatever it is, 1 over f or not, is not enough to explain the avalanches. That's one thing. Another thing, it sounds kind of trivial, but try to see what happens when there is no human in the MEG machine. It's not so trivial, because the MEG sensors, they have some correlations. They sometimes detect events that happen in the void. I don't know what that is, but I mean, they detect some electromagnetic events, and they have correlations, even in empty scanner. And here, at least, we see that this behaves more like an exponential for the avalanche analysis. I just chose randomly 8 data sets of humans, and I overplot them here, and the blue are the empty scanner data sets. There is another analysis that claims to be a probe for criticality that was published in 2009, and I repeated the same analysis, and I just got the same power laws for both. They also get some power laws, but of different things. It's not avalanches. These are periods of synchrony. And you just get the same results. I can show you these figures later. So you need to do this kind of thing.
Isn't the temporal information also important? Which size follows which size or something like this? Or just distribution sizes and all this?
It's a good point. To first approximation, you can assume independence, but we know that there are some temporal correlations, but really they are weaker. Like typically after a large avalanche, you're not likely to get a very large avalanche again. Things like this. Another thing is to convince ourselves where the cutoff in the power law comes from. So if you just take subsections of the array, for instance, the central 34 sensors or 68, 136, I just multiply by 2 each time. So you see that the cutoff is just earlier if the system is smaller. The slope remains the same. Another analysis that we did is to use some coarse grain sensors. The idea is that I group 4 sensors together, and then if one of these sensors was active at a given time, I say that the whole new sensor was active. So I get these kind of coarse grain sensors, and the analysis was inspired by the ideas of renormalization group theory in physics. The basic idea that critical systems are supposed to be self-similar. If you zoom in or zoom out, they should look the same. And we get, again, the power law with an earlier cutoff because we have fewer sensors.
Did you try cross-graining with this empty scanner?
No, I didn't, and I can try. I can try as well as other types of coarse graining, because I used an OR operation, maybe a max or majority operation would show different things. But, didn't try it yet. So I want to go back to the effect of delta t, because so far I showed you some results. What we find is that for different values of this bin width, we still get nice power laws. It's not that we don't get power laws if we take other delta t's. But but the exponent is affected by delta t. So this is the same human that I showed you, and that's the best fit. By the way we fit the data is using maximum likelihood estimation. I just take all the cascades. So I have, say, a few thousand cascades. I assume that this sample comes from certain distribution, like a Paolo or an exponential. Then we fit using maximum likelihood. And I don't use any threshold. I just take all the events from 1 to the maximal cascade that I had. So now, if we look at shorter bin width, we get steeper exponents. If we look at higher bin width, larger, we get shallower exponents. The reason is simple, because if you take larger delta t's, you start to concatenate avalanches. So you're more likely to get large avalanches, and there will be less small avalanches, right? So you get a shallower exponent. So how do you choose? We know that it can be somehow related to the finite size of the array, but we don't really know the conduction velocities and all this, and it's kind of complicated. So we need some external source of information to say something about the delta t. I mean, one way is to start looking at the inter-event intervals. If I take all the single events, all those events, not the cascade, it's just the single events, and I look at all the intervals between them, between consecutive events, and I plot the distribution. I say that it doesn't really have a scale, and it's not easy to fit it with a certain known distribution. For instance, in the cortical cultures, life is easier because you see a clear separation of timescale. You see a bimodal distribution, very short intervals within a cascade and very long intervals between cascades. So it's kind of easy. You put your delta t somewhere in between. But in the context of intact animals, in particular the human brain, you don't really see that. So what we do is we go back to this guideline framework of the branching processes. And we attempt to extract each time the branching parameter that describes the amplification in the system. So we just take each cascade, we take the first 2 bins in the cascade, and we calculate the ratio of descendants to ancestors in each cascade. And what it reflects is that after a silent period of size delta t, we had some events that perturbed the system, and this was the amplification in the next bin, again, of the same bin width delta t. So for each delta t, I get 2 numbers. One is the exponent of the Paolo. The other is the mean branching parameter across all the thousands of avalanches. And then I do a phase plot. This is what I'm going to show you now. I plot the exponent as a function of the branching parameter for different values of the bin width, delta t. This is what we get. So each point here is an individual, one subject at a given color. Each color is a time scale. So you see 104 subjects, and again 104 subjects for a different time scale. And you see the trend. As you increase delta t, this is small delta t, the branching parameter increases, and also the exponent becomes shallower. It goes towards -1. But there is a specific time scale at which we get roughly 1 and -1.5. Let me show you also, this is the same, just the means and the standard deviations in each case. So it's a mean over the whole population for a given time scale, 1.67. And for 3.3 milliseconds, you see that it's consistent with this idea of the branching process. It's not a proof, but it says that at least for a certain time scale, we get that the branching parameter is 1 and the exponent is -1.5. If we repeat the whole analysis for different thresholds, I'll show you, for instance, a higher one, 3.5 standard deviations, you expect to get a little bit better signal-to-noise ratio, then we get roughly the same result. It's even slightly better, but the effect is not too big. So the results are roughly invariant to the threshold, and there are other data from monkey experiments showing very nice invariance for the threshold. So the results are sensitive to delta t, but we have this kind of way to pick up a particular delta t. Another thing that we can ask is whether the results reflect some signature of the individual. So what I do is I take the first 2 minutes in each data set of 4 minutes and the second part of the data, and I use each to calculate alpha. This is alpha from the first half, second half, and these are just the individual subjects. So we see that the results are correlated, and we get similar correlation for sigma. And I also analyzed 20 data sets from Cambridge. That they have another machine. We have CTF. They have Electa machine, which is worse than the one in the NIH. Now I'm recorded on the video saying that, but still, so you get poorer power laws, I must say, but still you get roughly the same behavior. But the advantage that we had there was that the subjects visited the facility twice, separated by a week or two. And we get the same correlation. So it's kind of interesting when now I think about something. I was wondering whether these correlations can be somehow related to, just if you place the subject in the MEG machine and the distance between the sensor array and the head is a bit different, it may also affect the results, maybe. So I thought maybe it could be due to that, but here they visited the machine twice, so no chance that they positioned them at exactly the same distance from the sensors. It depends on your head size and other things when you put the subject into the MEG. So it seems that there is a signature for the individual. And for instance, I would expect that people with a tendency to epilepsy would be, for instance, here. They would have higher sigma values at the right time scale because like slightly more than normal amplification. Last point that I want to say is about what happens when we break down the analysis to different frequency bands. Many people like to look only at alpha, beta, gamma, whatever. So here are the results when we just filter all the channels within a particular frequency band and compare to the broadband. So the gray curve at the back is the same in both cases here, is showing the broadband, what I showed you before, for the same subject, 1 hertz to 150 hertz. And here you see the low frequency bands, delta 1.524, theta 428, and so on. You see that you still get some heavy tail distributions, but the exponent can be very different. Only for the theta band, it roughly resembles the negative 1.5. But for the high frequency bands, you clearly don't get power laws. You get something which looks more like an exponential. So the main take-home message here is that this analysis reflects some kind of scale-free behavior in the system, and it's not the right thing, or it's a bit artificial if you try to break it to different frequency bands. Now, we did some analysis of, for instance, the event density. Namely, this is a grand average from, I just collect all the events in each spatial location from each subject, from all the subjects. So the hot area here reflects, they reflect where you get more events, the higher density of events. And if you correlate it with the gamma band, the power in the gamma band, you see a very similar pattern. And if you correlate it with the high gamma, even better. Why? Of course, the low frequencies are important, but the higher frequencies, they have very, very fast fluctuations, so they can contribute more events. In areas where you get more gamma or high gamma, you just get more fluctuations. You get more events, so you get higher event density. So those frequencies do contribute to the avalanches, but maybe the avalanches, they reflect some kind of crosstalk between frequency bands or some other things that you just beyond those frequency bands.
What?
Yes,
When you took
that's
different
it.
frequencies, do you take different delta T's, or the delta T's are all the same?
It's a good point. I use the same delta T, just the same. Just wanted to change just one parameter, but I agree. I can change the scale too. So just to summarize, we used resting MEG to probe for criticality, in particular for neuronal avalanches. The analysis that I described today is very similar to what people did with LFPs. And I think that the new things were mainly that I allow myself to take both positive and negative events, and that I use this method to also look at the branching parameter to select a particular delta T. So we got this power law behavior, but we didn't get it for phase shuffle data or empty scanner data, and we saw that we get an earlier cutoff if the sensor array is smaller. If we coarse grain the array, we again get the same results. So indeed, the exponent that we measure for this power law depends on the delta T, but at a given bin width we got roughly a branching parameter close to 1 and an exponent close to negative 1.5. And again, we got similar results for 2 different MEG facilities, 2 different machines, one at the NIH, one in Cambridge, and the results were consistent across 2 visits or 2 different parts of the same data sets. So I believe that we believe that this is the first time that people show neuronal avalanches in the human brain, at least in the large-scale human brain, and it supports the hypothesis of criticality in this specific context of critical branching processes. And this is nice because it tells us something. It supports the idea that propagation of activity in the brain can be described by a critical branching process. And then we know a lot about these kind of things. We know that we have some kind of scaling, self-similarity, so we expect to see avalanches at all scales. And if avalanches are meaningful, for instance, if they code for something, because here we looked at only resting state activity, it's very interesting to see if they code for something, then it means that you can extract things at high resolution, maybe when you have the LFPs from a dense electrode array, but you get even some kind of resolution, and you see some higher level reflection of the avalanches at the large scale brain activity. Now, it's more than that. Now, criticality is associated with optimal information representation. So you expect that some disorders in information processing may be reflected in deviations from criticality. And we can probe with this analysis. Even more specifically, I showed you that the avalanche analysis is sensitive to imbalance of excitation and inhibition. If you have too much excitation or inhibition in the system, you get deviations. So it can be a certain biomarker or a probe for these disorders that reflect imbalance or that are affected by imbalance of excitation and inhibition. I mean, and it's interesting because today we can affect the system even not only pharmacologically, even using optogenetics, like Ofer does, and we need some quantitative tools to quantify the imbalance of excitation and inhibition. Lastly, briefly, I'll tell you things that I'm going to do because this is like an ongoing project. There are many things that we can do. For instance, going into source analysis, because then you can focus on specific areas in the brain and do the avalanche analysis above a certain area, which is more relevant when you look at disease, for instance. Another thing is that we can reconstruct the underlying networks from the avalanches. Typically, what people do in fMRI, for instance, they just look at second-order correlations, and then they construct those networks. But we have the cascade. So when we have the cascade, we can infer, using some Bayesian inference, the underlying network. You get basically a probability above the space of possible networks, and you get weighted directed graphs. So you get the links, and you use some kind of higher order information, the cascades that are directional and run on the system. I think it's a very promising analysis because, and it would be interesting to apply it, but no one did much work in the context of the human brain with this approach. And we know that, for instance, in fMRI, Granger causality and other methods fail big time.
You're talking about a very large graph
Yeah.
here. So you get something about the local architecture, assuming that it's repeated. What is the magnitude of the phenomena like this? And what fraction of the brain is involved in each cascade?
In each cascade. I mean, it depends on the scale at which you probe. In my scale, I showed you that, for instance, you have a parallel behavior, and you do have some cascades in which you see 270 sensors active. It's like the whole brain. They are very rare, but you do see them. Most of the cascades are very small, but you can see many cascades that involve 10 or more sensors. So it's an area of a few centimeters in the brain. So I also started to look into data sets from different types of patients, like schizophrenic, obsessive compulsive disorder. These are MEG data sets. But I also have, I'm going to get some EEG data sets about synesthetics, and I have some sleep-deprived data sets from the lab of Giulio Tononi. And here's an example, because they were obtained using high-density EEG. So it's a very similar condition to the data sets I showed you now. I have 256 sensors compared to the 273. sampling rate was 500 hertz, very close to the 600. So the binwidth, the minimal binwidth is 2 milliseconds. So if I take 4 milliseconds, I get very nice power laws with alpha close to -1.5, sigma close to 1. And lastly, I intend to look for avalanches in the context of task performance, whether they can carry information about something. Because we do see the power law behavior even when the subject performs tasks, but you see some differences. For instance, you tend to see more large cascades during the task performance. So maybe they code for more rare events, and maybe they carry some information. I mean, the nice thing about the avalanche is that they offer you a certain natural way to segment the data into cascades, which can be code words. Maybe they carry some interesting information. I continue my modeling work. Some modeling, I did some models when I worked with Chaim with rate models, and I'm extending them to spiking neurons so that I can see avalanches, maybe, because in the rate models you can see them. And lastly, there is a project of DARPA. It's a very ambitious project that we started to be involved in. They call it PI, Physical Intelligence. And the idea is to combine, to really have hardware implementations of something that look like a neural network, some spin systems. There is another system based on what's called atomic switch array, and other systems, maybe there is a chip that simulates integrate and fire neurons. And the idea is to embed some intelligence into it. They really wanted to solve interesting problems with stochasticity. It's quite ambitious, but they all believe that criticality will play an important role in that. So it sounds surprising, but maybe DARPA believes in many things. And I'm doing some data analysis, some avalanche analysis on results from those physical systems, as well as the algorithm work. And that's it. I want to thank my principal investigator, Dietmar Plans, and NIMH and DARPA for funding. And here's the group. Jeff was also involved in the MEG analysis. He's a graduate student that also works at Cambridge. Andreas gave some help with the statistical analysis of the power laws. This is Dietmar. And that's it. Thank you so much. Yes, the short answer is yes, but there is a catch here, and it's important to say it. Obviously, you have a finite measurement of the system. So if I simulate a model and I have access to all the elements in the model, all the neurons or nodes, then everything works perfectly. But what happens if I have subsampling? So I only sample from a subgrid. Then you can find that you get an exponent of -1.5 for a given time scale, but if you measure the sigma, it will be smaller than 1. It can be 0.5. So you get the intersection at 0.5. Why? The reason is that you miss many events. There are events that you just don't see. And then the estimation of the branching parameter is biased downwards. And I can see it too. If I take a staggered array, namely like a chessboard, each other sensor, then I see that I get nice power laws. They have the exponent of -1.5. The delta t that gives you it also makes sense. You need to take a larger delta t because you increase the spacing of the array. But if you now start to estimate the branching parameter, you see that you get a very low value. And the reason is that, again, that events escape, and you're not really able to measure it. So it's very important that the array will tile the whole system, that the events won't skip. This is why I use the coarse graining and not a staggered array. Yes?
Maybe I missed it. What is the significance of the value of -1.5 for the exponent?
The significance is that it's predicted by a certain theory. There is an analytical derivation that starts with certain assumptions and give you a certain form for the distribution. And in this limit of relatively large systems, you get the -1.5 exponents. The -1.5 is there, but you see that the power law is the dominant aspect of the distribution. So these results imply, not prove, but they imply that the underlying dynamics, there is kind of a generative model, the critical branching process. And it gives you some information, so it gives you some mechanistic insight, not just, some kind of metric that you can use. Yes?
You're saying you expect to see similar numbers if it was actually measuring neuron spikes?
If you do the analysis on spike data, it's even easier because you don't have to do the thresholding, right? You start with the spike trends. You just take all your spike trends, and you collapse them into one big spike trend, and you start to segment. But the thing is that you have spontaneous spikes. You have some spikes that just, happen, and they are not necessarily part of a certain cascade. So the thing is that you have lower signal-to-noise ratio when you use spike data. The NLFPs and the MEG signals, the discrete MEG signals, they already reflect quite a large group of synchronous neurons that are active. And if you just take spikes, it's more problematic. So the difference is that instead of looking for real silent periods, absolute silence, look for relative silence. You have to set a certain baseline that reflects the spontaneous activity on the array. And then when you see beams in which you see some low level of activity, you segment and start a new avalanche. That's it.
When you say that...
But there are papers that are showing avalanches in spike data from cultures, yes.
When you say that criticality is good for information purposes,
yeah.
it's a very ambiguous...
I agree. I said it's very broad. I can tell specific context.
Information about what, it's sensitivity more than information purposes. Or some...
One aspect, so for instance, in the project that I did with Chaim, I started with mutual information between input and output in a system. And we know that it can be the entropy of the output minus the entropy of what can be the noise. So if the system is deterministic or the noise uncontrollable, then it's roughly the entropy of the output. You can write it as a certain integral. And when you change the variables to convert it to the to speak in the terms of the inputs, what you get is that the important quantity is the log of the determinant of a certain susceptibility matrix, sensitivity, a Jacobian, which reflects the sensitivity of outputs to inputs. And this should be average over the space of all the inputs. So in this case, it's very clear that you want to maximize the sensitivity in order to maximize the mutual information or entropy. And then because critical systems are known to be very susceptible, the susceptibility diverges in principle, the system likes to operate under many conditions, not in general, near the critical point. Depends on the input statistics in general. But there are other properties. For instance, if the system can just be epileptic, it will not have a nice repertoire of representation. Either you get nothing or you get a boom. And if the system is subcritical, also you get activity dies out very, very quickly, so it's like a vegetative state. So the critical system has a larger repertoire, maybe like a parallel distribution of cascades that are ready to represent something. I don't know. These are just speculations, need to be tested more carefully with real data, with behavior. Yeah. Okay.
Next question. Do you know anything about Mercury and how it influences criticality or cascade?
Mercury, like for instance, in the context of the story of autism and these kind of things? No, but so what you mean is like, for instance, takes people and that are
people that were exposed
to Mercury.
And Mercury, for instance.
I don't know. Give me some data, and we can do. I can analyze it before and after chelation, for instance.
We're talking about epileptic.
Yeah. So I'm hope to start soon to analyze epileptic data and see what happens there. I hope to get a big signature. I don't know. Yeah. Okay. Thank you very much. about the